Dear children, in the last module, we had learned that there are two types of cells, prokaryotic and eukaryotic. Now let us learn about what is a prokaryotic cell. Prokaryotic, it is derived from two words. Pro means primitive and karyotic means nucleus. So those organisms who are having a primitive nucleus comes under prokaryotic organisms. So prokaryotes have prokaryotic cells. And what are the peculiarity of prokaryotic cell? They have no membrane-bound nucleus and organelles. They lack a membrane-bound nucleus and organelles, and that is the first basic criteria of a prokaryotic cell. And which all organisms are prokaryotic? Bacteria, the BG or blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. All these are the prokaryotes, namely bacteria, blue-green algae, mycoplasma, and PPLO. They are generally smaller and multiply very rapidly than the eukaryotic cells. And they vary in their size and shapes. You can see the different types of the bacteria, the cyanobacteria, that is blue-green algae, and mycoplasma. Now, based on the shape, the bacteria, which is a first type of prokaryotic cell, there are four types. That's Bacillus, which is rod-shaped, Coccus, which is spherical, Vibrio, which is comma-shaped, and Spirillum, which is spiral-shaped. We have learned these concepts in the chapter Biological Classification. So cell organelles which is present in a prokaryotic cells are the cell envelope, mesosome, and chromatophores. They have a nucleoid, flagella, pili, and fimbriae, ribosomes, and inclusion bodies. So let's begin with cell envelope. It is chemically complex protective covering which is seen outside the prokaryotic cell and is made up of three tightly bound layers. The cell envelope in a prokaryotic cell is made up of three tightly bound layers. Which are those? The glycocalyx, the cell wall, and the plasma membrane. So first let's learn about the glycocalyx, which is the outermost layer of the cell envelope. Its composition and thickness vary in different bacteria. And it may be a slime layer, that is a loose sheet, or a capsule which is thick and tough. So the glycocalyx, which is the outermost layer, it varies in different bacteria and it is of two types. Either it is a slime layer or it is a capsule. What do you mean by slime layer? It is very loose sheet-like structure, and a capsule, it is thick and tough structure. Second is the cell envelope, which is a middle layer just beneath the glycocalyx. It is seen in all prokaryotes, and it is the cell wall which gives shape to the cell and provides a structural support to prevent the bacteria from bursting or collapsing. So following the first layer, that is glycocalyx, we have the cell wall in the prokaryotic cell. And following the cell wall, the last layer or the inner layer is made up of the plasma membrane. It is semi-permeable in nature. What do you mean by semi-permeable? It allows the transport of only certain substances through it. So it is semi-permeable in nature and it interacts with the outside environment. This is structurally similar to that of the eukaryotes. This structure of the plasma membrane in prokaryotes and eukaryotes is the same. So the cell envelope is made up of three layers. The glycocalyx, followed by the cell wall, which gives structural stability to the prokaryotic cell, followed by the cell membrane or the plasma membrane. Based on the types of cell envelopes and response to gram staining, bacteria are of two types. What is this gram staining? It was Christian Gram. He developed gram staining method. So based on how they respond to the gram staining, there are two types of bacteria. They are gram positive and gram negative. Who are gram positive? Gram positive bacteria are those bacteria which can take up and they retain the gram stain. And gram negative, they are not having the capacity to take up and retain the gram stain. Okay, so these are called as gram positive and gram negative. We can see gram positive is the Staphylococcus aureus and gram negative is the Escherichia coli. So two types of bacteria based on the capacity, whether the cell envelopes can take up and respond to gram staining. There are two types, gram positive and gram negative. So dear children, in this module, we learned about the first cell organelle, that is cell envelope, which is made up of three layers, that is glycocalyx, the cell wall, and the plasma membrane. Thank you.